Stefan Fried. Stefan is a fifth-year PhD student in the Department of Biological Sciences in the College of Science. His faculty advisor is Professor Sean Lee. Stefan will be presenting today on origami snowflakes, protein misfolding, and rare disease.
In this, the golden age of genetics, we've encountered a problem. See, in the year 2000, we decoded the human genome. We can now read all the DNA that makes us who we are, just like a book. If you read that book, it would be about 1 million pages. It would have 20,000 genes or so in it, and we now know of at least 4,000 mutations, that's small changes in DNA, that can lead to disease. We also know of 7,000 rare diseases, many of which come from one of those mutations. But all that being said, if you found out that one of your children had a genetic mutation, what would you do? It's a really difficult question to answer even today, and that's because the problem we've encountered is translating all this new information into real understanding of human health. And so we're trying to move towards that goal. See, a gene is sort of like instructions for making a very complex biological machine called a protein with a very specific three-dimensional shape. You can think of it like instructions for folding an origami paper crane. And so a mutation, much like a typo in instructions, may lead to a jumbled up piece of paper or something that looks still a lot like a paper crane. And when you think about the 7,000 rare diseases, it becomes even more of a problem. Not just paper cranes, but almost like origami snowflakes. Everyone's a little bit different because we can't go straight from instructions to final three-dimensional shape. They're just too complex. So what we've done to try to address that is to take some well-established biochemical principles and distill these down into simple questions that can be asked and answered of a computer in rapid fashion. We ask these questions to bring them to those principles together into a single distilled predictive score. This single number is predictive of whether or not a mutation will be harmful or harmless. And when we take this computational approach and look at a couple of rare diseases over thousands of mutations that are possible, we find that that single number, that single predictive score, can actually tell us whether or not that mutation will be harmless or harmful in some cases. It can also potentially tell us how severe that disease will be. But in some cases, we've also found that despite our understanding, mutations that seem like they shouldn't be a problem nevertheless cause really devastating disease in patients. So there's a lot more work to be done and a lot more ground to be covered, but using this simplified, distilled look at protein structure and misfolding, we want to bring together three communities. We want to bring together physicians who can be empowered with this simple number to tailor their treatments to each of their patients. We want to empower patients and their families to understand a little better about what's happening to them. And we want to enable scientists to delve into new areas of research about rare diseases that have never been studied before. And with these three communities working together, we want to make it so that when you ask that question, what do I do when I find out one of my children has a genetic mutation, you can have the answer. Thanks.
Thank you, Stefan.